Dr. Anna's Science of Light radio show. Today I have a most extraordinary guest, and I'm so honored to have Clifford Carnicom with me today. I want to say that he is one of the literally role models of what it means to be a scientist, somebody who has investigated a phenomenon that many people didn't want to know about. He calls it cross-domain bacteria. Others name it Morgellons disease. But it's done such meticulous research, and it really is a reminder for us scientists on how it's done. And I wanted to let my audience know that, when I first met Clifford and had an opportunity to speak to him about the live blood analysis that I've done in my office and the findings in the C19 vials that many of my colleagues like David Nixon have found, he was kind of reprimanding me a little bit in a very
Oh,
sweet way. And he was saying, Dr. Anna,
no,
you can't
no.
be saying what this stuff is, like graphene or hydrogel, until you have done the research. And I said to him, Clifford, I totally agree with you. The reason why I do is literally because I'm concerned that we're in the midst of an extinction level event where 5.4 billion people are injected with this, and I'm just endeavoring to get the word out. And so, from my heart, I really agree with him and his role model as a scientist of how it's being done. And I'm so honored, Clifford, that you're speaking to me today.
Th-
Welcome.
Thank you very much. Those words weren't directed to you personally, doctor, and I have a habit that I've been made aware of sometimes where I use the word you in a collective sense towards the general public and often professional communities. And I may have you made my usual slip there, so it wasn't directed towards you personally. But the point is actually certainly very well and still made. Absolutely.
Absolutely. And I think that, if we want to convince other scientists who don't want to look at this phenomenon, in particular of what I have found in the live blood, what others have found in the vials, and how it historically fits in with what you have found, I think is very, very important. And your history goes way back to the 1990s when you talked to the Environmental Health Agency. Can you explain to our audience a little bit about what you told me about really understanding the context of everything that we're finding, and what your experience was then with endeavoring to have your findings analyzed and reproduced by other scientists and sounding the alarm?
Well, the actual work does start about 1999. The story, I didn't know this at the time, but retrospectively, if we look at the actual story in my life and how it was going to affect me, began around 1994. It wasn't till 5 years later that retrospectively I became aware that circumstances had actually changed up in the northwestern US about 1994. So the EPA is a very significant starting point, I guess. The story is a long one, and I'm not very much of a bullet person in terms of speaking. So you'll have to just basically direct me and cut me off if I get too long, because everything is its own story. And there are many, many stories over 25 years. But a very crucial starting point certainly was the involvement with the Environmental Protection Agency. And the work starts because of modification of the atmosphere that is obviously taking place. And since I was a little kid, I would always be interested in meteorology and even aircraft. My father was in the service for 30 years. So I spent a lot of time outdoors, and it was very obvious to me that the physical world and the atmosphere was literally being changed, and it was being changed with machines, with aircraft. Okay, it's straightforward physical modification. So I started recording that process truly as sort of like a citizen journalist. This is when the net was really just coming into full strength. And I realized that as a citizen, I had the means to make information available. You know, it's pretty amazing when we look at the evolution and transformation of the internet. It's an entirely different world, right, in 25 years. But at that time, it was an opportunity for an individual citizen to say, hey, I'm seeing something unusual. I'm going to announce it to the world. And I certainly had no history, no tendency whatsoever towards, let's say, seeking attention or acting in any kind of public role at all. But it was rather profound what was being observed. And of course, you know, it's the reaction, well, you know, doesn't anyone care about this? I mean, this is the atmosphere, right? And so again, there's many things that happen in between, but if we say the work starts, and the work starts with the point of observation, that's always the beginning of the work. I do not confine the experience of this world to observation, but observation is a common starting point and meeting point for us. So I began the process of observation. I certainly had a background in the sciences, but it's also rather obvious and logical what needs to be done. So I start looking, and I start recording, and I start collecting. And in that process, I think it's certainly well within the first year, there were many things, of course, that were starting to be discovered, but there was an unusual physical material that was making its presence early in the game, about a year into it. And another standard I would set for myself is it's very rare that I would become, let's say, dedicated, might be interested, but I wouldn't be dedicated normally towards investigating a phenomena that was going to be requiring investment of my time and effort, unless I saw it repeating. Right? Repetition is one of the very first sort of conditions that I would normally set on how much time I would invest in something. So this material, this unusual material, it was airborne material, and it was filament material, appeared on numerous occasions, and numerous individuals were reporting this. As a matter of fact, I didn't have a sample of this. I'm simply paying attention and being aware that people are recording the presence of this material. And then I eventually do receive a sample of it sent to me on a case that was documented. And I had several individuals send this material to me. I'm looking at it under a rather primitive microscope, and I start subjecting to chemical tests. And from the very onset, it's an unusual type of material. The size of it is submicron. Ultimately, it's learned. Originally, I wouldn't even know that I'd get down to that kind of size, but ultimately, it's submicron material. And my frame of reference in comparison back then is, well, what's the smallest thing that concerns us in the environment historically that might be of a filament nature? And so asbestos came to mind. When I looked at asbestos, I find that, you know, asbestos was about, I think about 2 microns across. I started looking at all filaments, all natural, all synthetic filaments, and nothing was fitting. And nothing was fitting to the point where, again, this would be very unusual for me, but I, along with a colleague, we wrote a letter to the head of the Environmental Protection Agency. Carol Browner was the name. It was actually straightforward, again, based on logic. If you had an unusual material that had very unusual chemical behavior and properties already showing up, appearing in the environment in numerous locations, that is, first of all, it's not soluble. So if you have something that's going inside your body, it presents an obvious environmental or health risk. You know, if you're talking about asbestos fibers, right, that are hidden in your wall or in your brakes or something like that, if it's showing itself in a straightforward fashion in the environment, and it is airborne, and it's found on the ground, and the whole idea of spider webs, it's like we can't take the time to really entertain some of the diversions that were put in place very early in the game. But let's say I'm very much aware of the diversions, and believe me, it doesn't satisfy the spider web criteria. So a letter is crafted.
That's like the people that are telling us that we're finding cholesterol and salt crystals when these are very elaborate microchips that are evolving. I totally understand the diversion thing.
There is no end to that, and that's actually, unfortunately, consumes way too much of our time and effort, and we've lost a lot of time in the process. But that's a deliberate strategy. But a letter is sent to the EPA, and it's a very straightforward letter. From my point of view, it says, such and such is happening. We request you. It was an assertive letter, but it was not an arrogant letter. It was assertive, and it said, please identify the nature of this material. Straightforward. The EPA, what happens here? The EPA does not respond. That's the first thing that happens. They do not respond to the request, specifically of putting any effort of any kind towards the identification of the nature of the material. What they do is write back, and I believe it's several months. All of this is on the website. It's all documented, certified mail, the whole thing. I think it's probably several months elapsed, but nevertheless, the response was eventually to write back, but not acknowledge that they had received any physical material at all. It was a straightforward citizen response letter, and what their letter said was, how did that work? No, it wasn't at this point. Their letter was simply saying, we're not aware. Their phrase of choice was, we're not aware of anything unusual happening. That's what they did. Now, remember, I sent a letter making them aware that something was happening, right? But their response was, we're not aware. And they maintained that posture for, it was actually a year and a half. They had a couple of correspondences going on in between, and it's always this phrase, we are not aware. And I'm sorry, this is a whole story in itself, and I know it takes time, but if you keep me on here for 2 days, I'll do it. What happens then is that a Freedom of Information Act is filed by a third party. It has nothing to do with me. And the motive for that is a whole separate story in itself. But nevertheless, a Freedom of Information Act is filed to find out what actually happened here. It is at that time, a year and a half later, that the EPA, what they do is they return the material to me. Okay, they return it to me, and now their statement is, we have no obligation, this is paraphrasing, but the statement is, we have no obligation to do anything unless we ask for it. This is their statement. Okay.
And this is it's just incredible. And there's so many decades of your work. I have prepared a PowerPoint because I want to ask you some very specific questions about, because ultimately what I want to elaborate on is, what are some of the common features of what we are seeing in terms of the vials, and then now in the blood, with what you've seen all along. And so I think what I'd like to do is, I'm just going to share here real quick my screen, and I'm going to, are you able to see this?
Try to get a little bit more light. Yes, I can see everything fine at this point. Absolutely fine.
Okay, let me see if I can go full screen. Okay, so what I had what I did was, you wrote 6 papers in the past year that were quite extraordinary, because they were very methodical in regards to your methods and explaining what you had seen. So the first paper showed blood alteration and coagulation, which is a hot topic these days, because what we're seeing in the live blood analysis of vaccinated and now unfortunately in unvaccinated blood are clearly a rouleau formation, which has to do with clotting. And I personally have investigated shedding in people who are unvaccinated in regards to the effect of the vaccines, and found elevated D-dimers, which means that their blood is clotting. And the reason why I was so interested was this research that you did was pre-injection era. but there are certain commonalities here that I would love to talk about that may help the researchers now to maybe find out more about what we're really dealing with. And you named this thing a cross-domain bacteria. And what that actually means is that there are several genre of how you can classify an organism, whether it's a bacteria, a eukaryote, and correct me if I'm wrong, there is one other
Archaea.
criteria.
Archaea.
Archaea, and you found that this thing that you classified belonged in all three, which would mean something like this is a new life form, supposedly synthetic, because before it's usually you belong in one of the three but not in all three at once. Did I say that right?
Yeah, I think that's absolutely fair. By the way, not to belabor that initial story, but couple things on my mind in terms of what this whole time that we're spending together is about. Number one, that filament story that I told you about, that carries forward 25 years. That story didn't end in 1999. That story has carried forward and affects literally all of us every day up to our point in time right now, and continues to. So there's nothing about that being in the past. That would be number one. And then number two is, one of my main objectives here with our time is to attempt to communicate that for us to understand the current environment we're in, call that the COVID environment, this new era of literally almost mandated injections, this COVID area, to understand what is happening, transpiring in the COVID area. My contention is that we must equally understand, become aware of this history of 25 years that I have been involved in, because there is a continuum from that day that I started to describe to you involving the EPA's refusal to today. And I believe and think we will be very, let's say, limited in our awareness and understanding of what is happening in what we might perceive to be the COVID era, and that we must extend our time of reference to the first signs of, we could use this as a standard for the first time that something unusual appeared in the blood, right? And guess what? That goes back also to 1999 to 2000. And unfortunately, for whatever reason, we've missed that continuum that exists. But you will find that my blood was a topic of mine almost from the very beginning. Okay, so that's important to establish that reference frame. Now, in terms of this particular phrase, even cross-domain bacteria, that's a phrase that I coined. I coined that phrase because I had to, not because I wanted to. In fact, for years I held off. I kept saying, let's identify it properly, put it into the scientific, let's learn what we have. And of course, in that process, you would evolve a proper name for it. That process would never take place, no matter how much it was requested. Over many years, I was forced to, I didn't want to do this. I was very hesitant. You don't want to go naming something from an individual point of view. Even this term morgellons, these names cause nothing but havoc down the road if they result by spurious action, I guess I would say. So, but I did. I coined the term eventually, and I wrote the paper on that. And I told people I'm reluctant to do this, but I must come up with a term because we need something that is more representative, or at least fairly representative, of what the facts appear to dictate. And that is the identification of a biological organism. Everything says that inherent in those filaments from the very beginning, inherent within those filaments is the existence of a novel biological organism. And I coined the term cross-domain bacteria because that is the best term. And we know what's interesting too, when I coined that term, I never had heard of it. It just seemed to be the best I could come up with to describe what happened. I learned like within a year of that, that term actually existed. So there is a place where that term was used, and it had to do with some plant pathology. So that in the end, that term is not off the wall. It may be rarely considered or used, but it's a fair term. And as you described it, the qualities of this microorganism is that it does not attach itself singly to any of the classic what are called domains of life. The characteristics of it span the traits of essentially all life, and those criteria are based on genetic. Those domains are based upon genetic heritage. So yes, that organism, the term and the nomenclature was derived out of necessity after years and years of non-response to appeals.
And what you found in the blood, because you looked at people's blood, is very similar to what we're seeing now. For example, here I captured an image where you had looked at the cross-domain bacteria in the human blood and saw that very quickly it created this rouleau formation if these things were present.
Yeah, I would say the conditions, the observations are actually identical to what I'm seeing. They don't even appear to be just similar. They appear to be identical in terms of the general characteristic and behavior that's taking place. Now, the reason that these observations took place, the Carnicom Institute no longer has a laboratory. That's the case of about a good 3 years ago. I had about a 3-year period where I had to sit in basic wonder about the state of the world. I no longer had information. I had been looking at blood for 2 decades. And the things that I'm speaking of, they go back to 2 decades. It had nothing to do with the quote vaccine era. But I had about a 3-year gap that happened to fall at the time that the COVID era was appearing. And I had this 3-year gap where I had no information about the state of blood. And so I was simply curious, because all the time I'm curious, but I was curious, we have this major change in the entire world. And now we're talking about direct modification of the human physical being. This is something direct. And everything that I had talked about and do my work on was environmental, essentially. So I was curious, is there any change in the state of the blood from the time that I knew it over 20 years versus what was happening in the COVID era? I mustered up enough equipment to begin at least the process of observation. And what happened at the beginning, this is why we already have knowledge that it looks like something significant occurred at the appearance of the COVID era. This gets a little slightly tricky to sort out, but it's to be understood that the samples that I looked at, the individual, they were not vaccinated. Okay, they were not vaccinated, but nevertheless, there was something unusual that was happening when I just started the question of, hey, what does blood look like? And that is that slides were not being prepared with the same ease that they had in years past. I made dried prepared slides for years and years on end. So I was familiar with that process and how it was working. And what happened is those slides were not being made with the ease. The slides were not coming out clean. And you essentially had coagulation and such taking place immediately, where the slide was not even recognizable. The traditional slide was not recognizable. This is why I went to live blood analysis, okay, because I needed to see what was happening in real time. So remember, I have no tie directly with the COVID era at this time. I simply want to look at blood. But what's happening is the blood is looking different already.
Yes.
Than my previous work, and the only environmental change is the onset of the COVID era. But also understand, my work did not even introduce the COVID era, at least directly. May have introduced it indirectly, but the individuals that I looked at were not vaccinated. Yes. So here we have a situation where a phenomenal event is taking place, and that has no direct, and I have to emphasize the word direct, connection with the vaccine issue. But believe me, it doesn't mean it doesn't have a connection.
And then you had, in your second paper, you were really explaining the means and the methods of some of your analysis, which is so of interest to me now because we're endeavoring with what we're seeing in the vials with David Nixon. He's doing experiments with, for example, Wi-Fi and sees that these ribbons are exploding in growth as well as the crystalline microchip structures. And then if there's a Faraday cage around them, the growth is stunted. There's a assembly and disassembly process. And what's interesting here is you've very meticulously looked at investigating, well, what are you dealing with? And you've used all kinds of methodology with DC and AC voltage, and these are very sophisticated types of evaluation: near-red spectrometry, microscopy, visible light spectrometry, electrochemical analysis, enzyme analysis, magnetism, protein detection. And you wrote about this, and you have a very interesting laboratory filament report. So these were these filaments that you investigated, and you did a heavy metal analysis. And the reason why this is of huge interest to me is because what we find, for example, in Mike Adams's evaluation of the blood clots of the deceased who were injected, he found a carbon-based polymer with a lot of metals. And for me as a treating clinician who's finding these very strange ribbons that look just like what we find in the vials, EDTA, which is a chelator of metals, as well as it dissolves hydrogel, has been very useful in clearing the blood. So I captured this analysis, which I find extremely important. Can you speak to what you did here and the findings in regards to these very high level of metals that are clearly sprayed through geoengineering on humans, like aluminum and barium and others? Can you talk about that?
Sure. And this is another classic example of the thread of importance in the existence of these filaments from their onset in 1999 to the present day. I don't remember the exact date of this report. It would be easy to recover, but we know that it would be several years past. I'm going to guess somewhere around, I don't know, maybe 2016, 2018. Now, this report is actually very interesting, as many of them are. I have false reports that were given to me also in the history. This is a very interesting report. First statement would be that I did not conduct this laboratory test. This test was done by a top-notch environmental quality lab, and I'm even hesitant to say the state, to tell you the truth. It took me, altogether, it took me 2.5 years to be able to release this information. I had an arrangement done where a viable, appropriate, and proper sample was given, and you would call this citizen sponsored, but not by me. There was an individual that had this in. Like I said, this material shows up really, it's global, is what it amounts to. And they had an arrangement with this particular laboratory to have this test done. This is a high quality test. It's industry standard ICMP, inductively coupled mass plasma, something I would never be able to touch with my laboratory. So first class, millions of dollars of equipment involved. I did visit the lab. I visited the head of the lab and did this work. We talked about it. And so he does this work, and we had the discussion, and the trickiest part of the whole thing was, in his mind, he's saying, well, I know these things shouldn't be here. And I'm saying, can I make this information available to the public? And let's just say the answer was no at that time, because the liability and risk to this person's laboratory was strong enough that this person did not at all feel comfortable making this information available to the public. Well, let's just say his response was, I certainly don't expect to see this. And you can also ask, what the heck are these filaments, right? Where are they coming from? It goes back to the EPA. So I can't release the information. And it took me 2.5 years of negotiation with the parties to be able to release the information in a way that would not be interpreted as, let's just say, no identifying information is made available intentionally on this, but the lab is a straightforward transcription of the results. So this was first class laboratory work. And, you know, when I put up something like this, I keep expecting, you know, none of this is about me. I truly wish that we had a motivation to actually like ask questions, to seek answers. And when you see a report like this, that's top quality, and now you have the actual sample that's in question for the EPA, same type of material the whole time, and you get these results, and like I expect some kind of reaction. Right? There was no reaction. I think you're the first person I know of that is actually now calling attention to this report. All right. Nobody, I'm sorry, nobody did anything.
Clifford, I understand that so much. I mean, look at Mike Adams, who evaluated those cadaver blood clots, something that affects the entire world, and nobody has reproduced these findings. So it's just phenomenal. But I think it's so important to now discuss what you have found, because it's the correlation, exactly the historical reference frame of your work to what is happening now for any clinician, any researcher, is so important. So I can't tell you, you know, how much I think this is valuable information. And I mean, look at this aluminum and barium and these things that we know are chemically being sprayed on people, and that it's actually part of this filament composition. What did you think when you saw 12,300 of aluminum here? And I mean, these are conductive metals: titanium, vanadium, zinc. What did you think about this?
You have a construct, is what I think. Okay. You have a construct. You have something that is made. You can play all the games you want with trying to pass something off that it's not important, that it's just normal as can be, and somehow it's natural, even though, okay, I'm not going to really define what that means that I'm saying. It's a construct. This is
it's synthetic. It's either man-made or made by someone, but not by nature.
It is a constructed material. It's by design. You're talking about a sub-micron filament that has, you know, this is one side of the story. This is the inorganic aspect, and one inorganic aspect of this. You have an entire organic aspect that we won't even be able to hardly touch on. But when I told you this when I was speaking to you, but so I've mentioned that this filament, its origin is down to a submicron, right? Well, it's not the filament that is the story here. It's what's going on inside of the filament. Okay? Within the submicron, there is an entire biology that's going on that is, let's say, ill-defined at this point. I've certainly done the best that I can to go after it, but you need resources, obviously. You know, I was thinking, you know, people criticize, well, why don't you just figure it out yourself? People don't have the resources to talk. The things we're talking about, you are speaking at the nano level. I can't get at the nano level. I can only get at the micro level. Or ironically, I can get at the atomic level with infrared, but I can't get at that nano level, which is a very, very important and interesting part to be looking at. But my reaction is, this is a construct, okay?
Yes.
This is systematic. This is by design. This is by engineering. However sophisticated that is, it's still a design.
Absolutely. And, you know, I've been talking with Karen Kingston a lot about synthetic biology, synbio, artificial intelligence, the gain of function of merging biology like bacteria, viruses, with artificial constructs. And so this paper that you wrote is one of the most concerning to me, as well as one of the most important findings, because one of the concerns that I've had is in the live blood of the structures that I'm seeing now is there's clearly a response to external Wi-Fi radiation. At least we don't know what other things. It appears that other researchers who've called some of these ribbons graphene-like structures, which they may have carbon in it for certain, but that they are responsive to things like ultraviolet blue light. The reason why I found this interesting is so you applied AC voltage, so an electrochemistry, and looked at what happens to these filaments and the surrounding blood. Can you kind of comment on this here?
Yes, thank you, Dr. And thank you also for graciously moving me forward just a little bit at a time to each paper, because yeah, they are profound. There's no way around it. And I'll try to at least hit them for a couple of minutes each time. This is a profound finding. There is actually no way around it. And it's also an excellent demonstration of the marvels of science and how it works, because the result that was found and published in this paper, this was never part of the plan. It's not what I was doing, okay? An extraordinary set of observation arose, but it wasn't in any way at all what I was seeking. What I was seeking was the fact that I know that electrochemical methods can be used as a method of inorganic analysis. That's really important, because I don't happen to have an ICMP machine with billions of dollars, but I was able to get access to electrochemical equipment. It's still not, let's say, free, but let's just say it was within my range. So I have a means of inorganic analysis, which is very slow evolving. So I simply wanted to analyze the blood from an inorganic standpoint. And electrochemistry is simply a tool that was no different than if you were, say, going to use a microscope or infrared spectrometry. I just wanted to use the tool. And so I started using the tool in a way that would be, proper to start to get some information, and something extraordinary took place. And I didn't know how extraordinary it was until I started looking at it further. But the straightforward and simple reaction is that there was a physical transformation. There was a physical process taking place in the blood with the application of what it ultimately is a very low current. We're not talking about high currents. I believe I'm in the 1 to 2 milliamps. It might be in the microamps by the time we're done when we consider dilution factors. But a physical change of the blood was occurring. Now, that by itself may not seem so unusual, because I've done electrochemistry work, straightforward studies, and you will have transformations. That's what electrochemistry is largely about. So maybe that's possible. But first of all, this is blood. This is not a salt solution, which would be a classic case. This is blood. And you had the appearance of physical forms that they weren't expected, but again, I don't know what things are until I look. So I saw that there was a transformation taking place. You actually had two different layers that would form in the blood, one being a frothy, foamy material, which I actually started to study that. And what does that mean? Because foam and froth in our classic lives usually often means pollution. But I found out, guess what? There's another equally likely explanation, and that's the existence of proteins. Proteins generate a froth and foam material too. So you had a foam and froth material at the top, and you had you'd have a settled later. In other words, there was a gradation by density. So you had a couple layers form, and then you had the material in between. So where does the big surprise come in? The big surprise comes in when you start looking at this material.
So Clifford, real quick. So, but what you saw is you applied an electrical voltage, and these filaments started growing rapidly, correct?
What happens here, the extraordinary observation is that the application of electromagnetic energy to a vial of highly diluted blood literally caused the formation, the development of the very microorganism that I'm speaking to you about that goes back again, all the way back to 1999, which I spent years developing cultures. I mean, literally years and months of different scenarios to try and create a culture. Here, within a period of roughly 2 hours, never seeing these formations. I looked at the blood before. I showed you the pictures of it, you being the collective public. I showed the photographs of the blood, right? You don't see what I'm talking about here in that blood. You see coagulation, right? Yep. We're saying here that an observation is recorded where within a couple of hours, you have a physical transformation of the blood that takes place, and you have the development of a micro form of life. It is a form of life that's been studied intensively. Remember, what I'm seeing here is not a surprise to me in terms of its history, but it sure is a surprise as heck to see this forming in a couple of hours with the application of a specific type of energy. So this is the observation, that it forms in a brief period of time, and it forms at a highly sophisticated level, at the highest level of manifestation of what this biological organism is capable of doing, at least in a cultured environment.
And here is where I want to interject and remind people of what we've been seeing in the vials as well as in the blood is a form of self-assembly of growth from a nano to a micro scale in a short amount of time. And there is again a parallel that I want to just allow our listeners to see towards, the research that I've done and the research that you've done. And so here we have a situation where he applied an electrical current and you see a rapid growth that's never been seen before in other cultures, but clearly there's an external force that can accelerate the self-assembly of this synthetic organism, and it changes the blood, surrounding blood that is exposed to it. This is a highly significant finding because it really says to us of what we're finding in this time that we need to perform similar experiments with Wi-Fi, with electricity, with light, and see how is it affecting the growth of the structures that we're seeing.
Absolutely correct. And I'm very grateful that for the first time that I know of, and this is to you specifically, that a connection is being made that gives us a broader framework in which to interpret the changed world, the altered world that we are now, I must say, subject to. I can't just say live in, that we have been subjected to.
Absolutely. And to me, understanding the broader framework of this transhumanist agenda, and that really everything in terms of transforming the human 2.0 to this synthetic biology has been going on for more than 20 years. And it's been going on through the atmospheric geoengineering, the change in the entire natural world who's been sprayed with this stuff and transformed. And we are literally, as Ilana Freeman would say, in the final stages of this transformation, and we're sounding the alarm bells because finally, we're seeing this thing, and it's like, oh my God, this has been going on for a long time. I want to get back to this particular filament because this is so much of what we're seeing right now. Again, it's a low-level electrical current, and you see how the blood transformed into a very linear field, strange agglomeration with these things. A very, very interesting findings.
And this had happened actually right. So here you have another remarkable observation, right? And that is the alignment. You have field alignment of some type taking place. You don't have to know or talk about something to say what it is, but you can describe it, and you can acknowledge it as being unusual and extraordinary, and then that sets out your job that you need to do. Here you have the apparent field alignment of materials. Now, I would say, though, from what I know, that those are more likely protein, at least dominantly protein materials, but you have an alignment of them. Now, what people don't know is that I've seen the same alignment several times in the past. Years ago, I've seen this alignment under the scope. And the way I do my work, like I said, unless I can repeat it and basically I have control over the environment, I simply register it and I say, you know, that's interesting, but I don't have full control over that environment yet. I don't know how to replicate it. I saw this many years ago. And what happened was I didn't post anything, okay? But you can bet I registered it. And when I saw this time, years later, I immediately recognized it's identical. It's not similar. It's identical to what I had seen before. The difference now is I have a mechanism that's been identified that appears to have caused this. And this was the application of energy, electrochemical energy. And so now it is to be recorded and released. But once again, that history goes back years before also.
So I just want to interject here some images from my office. This is what I see in unvaccinated blood, this rouleau formation and these structures, whether or not this is environmental from geoengineering, whether or not it is from shedding, but it is present now, not just in vaccinated, but also in unvaccinated blood. Highly, highly alarming to the future of humanity and what this means, because clearly the health effects of such a rouleau formation, a stacking of inability to deliver oxygen is very, very concerning. And I just wanted to interject that here. These types of ribbons like this have been found by many researchers also within the vials. They've been found in masks, in PCR swabs, in rainwater, very similarly looking. And it's just interesting that you've seen that a long time ago.
Yeah, there are many interjections that can be made because there are many topics that have evolved over the years. And, you know, if we accomplish the fact that people will consider the entire body of evidence and information that is available to us, you know, believe me, I don't have all the answers at all. As a matter of fact, I have probably 100 to 1,000 questions for every single thing that I'm posting. But we at least have a foot in the door to know that we don't want to falsely assume that something starts at such and such a certain date when such and such thing happened. We have a very broad frame here to work with, deep, affecting the entire Earth, not even a single nation.
Yes, absolutely. And here are some images of this presumed protein. And again, I just want to show something from my office. I haven't analyzed this, but when I saw these images of this protein, it's an unvaccinated patient, you can see how abnormal the blood is. And this is someone who has symptoms of what's called long COVID. So we presume. I just find it very interesting because when I looked through your work, looking at the similarities of some of the images, I was just stunned by that. And what I want to what I want to switch gear to now is something of high interest to me, because in the C19 vials, we know that there are components that are creating hydrogel. For example, the polyethylene glycol, there is the SM102 in the Moderna shot. And it is part of what we believe is this self-assembly mechanism that is clearly affected by frequency, Wi-Fi frequency. It is a carbon polymer that has metals in it that can grow pretty substantially. And my concern is that it is part of this clotting mechanism that we see. And what I did here is I took one of your handwritten notes, and I am so happy to see that somebody has similar handwriting to me, challenging to read. But what you had mentioned here is that polyvinyl alcohol appears to be within the example a very important component of these cross-domain bacteria. And I just want to explain that that this is an essential part of hydrogel. I snatched this from Wikipedia, and polyvinyl alcohol is a synthetic polymer that it has been used in a variety of things like 3D printing. It's part of biocompatible hydrogels and has been used in applications like vascular stents, cartilages, contact lenses. And in medicine, it is used, for example, here, hydrogels are used in insulin delivery system, medication delivery systems, and again, in biomedical applications. And the reason why this is so important to me is also that this is from Ilana Freeman's book, Geoengineered Transhumanism. Polyvinyl alcohol has been used in what's called the M spray. So this is related to the smart dust or neural dust that has been sprayed or has been used in the warfare application against humanity. So polyvinyl alcohol's iron and gluten is, when heated, can turn any object into a robot and controls their locomotion. And neural dust replaces the wire electrodes that are used for the brain-machine interface, and remotely directed by ultrasound can monitor nerves, muscles, organs in real time. And so once it's in the cortex, it is interrogated and energized. So the reason why this is important is these components of hydrogel that you found in these cross-domain bacteria, or however we want to call them, that are in the vials, have also been sprayed on humanity, and they're part of the infrastructure of control that is transforming humans into these hackable animals, and that is already happening everywhere. And so I just wanted to mention the hydrogel connection. Do you have any comment on this?
Yeah. This is how we met, first of all. You are the first person that I saw recognize the significance and the connection between polyvinyl alcohol and hydrogel. We should be fair about how I did my work. When I did my work, and I mentioned to you, I had no idea the term hydrogel. There's a million terms I don't know. I didn't know anything about hydrogel. What I had was an infrared instrument, and I knew the value of that instrument was its ability to identify what are called functional groups within a chemical compound. I spent years using that instrument. The public has a library now of at least 1,500 spectra that are a window of opportunity and understanding for us to specifically identify chemical constituents and nature of the very materials that we're speaking of. But I did my work on the pursuit of what are called functional groups. So for years and years, I'm trying to identify functional groups. And you'll see in my notes, I think, I'm trying to figure it out, but I think the notes actually will go back to 2014, and maybe we're going to find something at the very beginning. But at least 5 to 7 years prior to the time that you're even showing this one, I already had these appearances of this particular functional group that was catching my interest. And so I remember reading about polyvinyl alcohol, right, or trying to learn about it. And there was no other conclusion that I could come to very early in the work through a combination of methods, not just this, that polymers themselves in general were at the heart and foundation of much of the growth. I saw materials form that truly had the appearance of what you would call like plastic. Okay, I had very phenomenal things happen. My papers were only a fraction of what was actually witnessed and seen. The laboratory notes do tell quite a bit more, but polymers were there from the beginning in so many ways. The culture work eventually splits up into 3 different types of metabolic products. One of those products is a water-soluble protein. I talked to you about it, what's called ATR infrared, which is a plate. You put on your sample on a plate, and it's much easier to work with, so it's getting rid of the water. Continually, I would see this film that would appear, okay? And it had the appearance of like a plastic film, okay? Like a shellac film. This stuff was going on for years, I have hundreds of infrared analyses that we're going to find this. If we're willing to go into the library, we're going to see that we have chemical signatures of all these things that I was working with. And polyvinyl alcohol is one that arose repeatedly. And then I remember encountering that, it was stating, well, this is a synthetic material. Well, what does that say? I'm finding it inside after breaking down a specific microorganism. I'm finding this within the metabolic products of that life form, right? So I'm glad that we've made the connection, because to my knowledge, you're the first person that is able to put this in context. Once again, we have a history of years and years indicating and showing us. I did other work. Sorry, I get carried away here, but I remember that I did growth studies, because I have video actually of the growth of these filaments, a time lapse. And I remember I was asking a question in my mind, and this is, I see this growing in the culture, and I can record it with time lapse photography in a fairly short period of time. In a few hours, I can show that this is growth. And so I started asking the question, well, what does that mean in the human body? And I took that culture work and I extrapolated it to what it would mean to grow in the human body. And I had studies that were proposing, the growth of this material in the body and how long it would take. And all I remember, I was dealing with things that, even though you think maybe it's only a culture dish, I knew that within months to years, I could have, let's call it extensive growth in the human body of a polymer material. Okay? So I basically transferred it into the human body, and it's like, guess what we're seeing at this point? What are we seeing at this point?
We're seeing those large clots, and I just want to interject here.
Right.
My research was from the perspective of what's called, there's a book called Nano Brain, the making of an artificial brain from a time crystal. And when I looked at the materials that were used to actually create an artificial brain, it was hydrogel together with carbon nanotubes with certain metals insertion. And it clearly showed that this material was able to grow from a nanoscale to centimeters long in minutes under certain, energy exposures and frequency exposures. It was self-assembly, self-learning. It had the capacity to basically create an artificial intelligence supercomputer that would simulate a human brain. And when I really looked into the potential implications of this, how would you hijack human consciousness if you wanted to create a generation of humans that were no longer conscious but can actually be uploaded in terms of consciousness to the cloud. I was very concerned about this, and there's so many applications to this. This is why I love talking to you, because you've looked at such broad aspects of this. And in my research, from a different perspective, it's just kind of putting pieces of a puzzle together. What I wanted to get to, which I find absolutely extraordinary, none of us have looked into the ribbons close enough with high enough magnification to find what you found. But somebody had sent you these morgellon fibers. They were called morgellon, so they were similar to these cross-domain bacteria life forms, but you actually found blood and DNA inside of them. Can you speak to that a little bit?
Yes. Here's what I would say, and that is another important stage in the work was, you have the appearance or onset of a condition that's given a certain name, right? The name is given morgellon. It's the same thing with language. Language is a problem. That whole language is a problem. Okay, but nevertheless, it was given. My work, you will see, it isn't too long before I start saying, this is not as it is being portrayed to be. It is not just a skin condition that affects a few people. Fundamentally, this is actually an assault on the human body, and it affects the entire body, and I call it an assault on the human body. has nothing. It's totally in error to regard it as some kind of skin condition. But nevertheless, it was given this name. Now, of course, I have. By the way, there's about 400 papers that have been written roughly, so that's where the history is. Okay, all the work is there. But we talked about the environmental film that the EPA refuses to identify. Then mid-level, you have the appearance of a physical sample coming out of people's skin or on their body that has certainly the superficial similarity, but that doesn't mean that much until you start doing analysis. And when the analysis was done, you will find there's a point in the papers where I will say a match. And then you're going to find papers that say a second and a third and a fourth and a fifth match, because I did it over and over and over, as I usually do. But the fundamental conclusion was that the nature of the material that is being reported in these biological samples is identical in all the ways that I can use, which are quite a few. Again, heavy duty infrared work all the time, but is identical to that of the environmental filament that we spoke of in 1990. That is a crucial stage, right? Because now you have something that is making its presence in the body that has a direct connection to that which was refused to be identified years prior. So that's a fundamental discovery. Now, you're speaking of a couple of things. There's a distinction I'll make, but in the what you're speaking of on the right side here, the left side is a biological sample. The right side was a environmental sample, and I told you this story of how I had a health professional that said, "Clifford, I'm seeing some things that you can't see with your equipment. Would you like to visit me?" And that right side here is a photograph with a session with this individual who was, let's say, highly accomplished in the field of microscopy. And by the way, he had never, he had never seen this particular development, or not at this level. But we spent a session under the scope, and he was after showing me that there were biological structures, and this is from his assessment, not mine. He was the one that was calling them erythrocytes. He was calling them, he was seeing what his best analysis was the equivalent of a freeze-dried erythrocyte. That's what our meeting was actually about. In the process, again, everything has its own story, but in the process, he sees this for the first time, and this is a congregation, a conglomerate of cellular structures. Okay. Now, there is no end story here because the work was never done. Right? There's so many things to do, but you had the appearance of a conglomeration of biological structures, and it's not going to. Maybe blood is involved here, but that's not the point. I don't speculate because there's no purpose. There's no purpose in speculating. That's the extraordinary observation, because this occurred within the filament material. This was the very portion of the sample that went to the EPA. And what was this discovery? This discovery was that within the environmental samples, there were biological structures that existed, right? And this goes back.
Mind-blowing. So there are filaments raining on us that have blood cells in them. That is like phenomenal.
People will ridicule this stuff, but it's like, sorry, we got to deal with the business at hand. And sorry, if you get lost, you get left behind, because whether we like it or not, these are the findings. My observations they seldom have anything to do with me. It has nothing to do with me. It's an observation, and it's a record of what happened. This is a record of what happened under a highly skilled a professional with very high capable equipment that recorded remarkable findings, and they were made known to the public in those dates, and you'll see it. And what is our consequence thus far? Well, guess what? We may be at a point where we're going to go back and we're actually going to revisit this work, hopefully with your help and your connections, because that's certainly what we need to start to gain a broader understanding of what we have. Now, I have to make a correction or distinction in that in the DNA issue. The DNA issue is not anything that was ever seen by someone else. I don't know of anyone besides myself that has approached the DNA issue. What happened is in my culture work, you will find, I think, 3 different records at different times, separated by several years. I kept returning to the problem several times. I had this question that arose, and the question is, you know, everything to me says this is a biological organism. It might be a very unusual form of life, but it is a form of life. It does reproduce, it does grow. It follows the natural laws of biology that I can see. So that means it should have DNA in it. If it's alive, it should have DNA. So I took on the question, can I extract DNA from this organism? And the answer is yes. I've done it 3 times. I also realized my limitations. Extracting DNA is not sequencing DNA, but it's still beneficial. And I needed to prove for whatever reason, many reasons, it's part of the validation of it being a life form or not. But on 3 different occasions, I have extracted DNA samples from culture work from this organism.
And what is important to understand in regards to synthetic biology, that DNA sequences can be modified through gain of function to do specific things. For example, the spike protein sequence has a sequence that is producing hydrogel actually at a pH around 7. So at the acid base equilibrium of the physical body, the injected spike protein sequence of the presumed mRNA that people got actually tells the body to make hydrogel, which is a plastic carbon-based polymer. So there's synthetic biology is the mixture or the alteration of natural DNA to make something synthetic. So even though you found DNA, this can still be a synthetic biology.
Absolutely. We are constructing DNA, right? We're at that point, right?
And what I want to show here is just a couple images, again, just to show our viewers. This right here is an image of what you had, a magnification of this filament. And what you're showing is this internal structure. Right here, this is somebody who has long COVID in my office. And this is what I can see in the filament structure. And then if you see here, this is 1,500 magnification by Mike Adams of one of the clots that he pulled out of the cadaver. And you see the central aspect here of almost a hollow tube-like structure that appears to be common. I just wanted to put this as an observation out there. This right here is part of your next paper, because you did many different chemical analysis. And what was very interesting is you found what's called halogens. So halogens are fluoride, bromide, iodine, and chloride, and other metals, for example, other components like nitrogen and sulfur. And so you did pretty significant evaluations, found large amount of iron, and actually had a lot of information regarding the overall iron metabolism and how that was contributing to some of the health aspect in the body, and then again how antioxidants were helping with it. So this is what you were talking about, the foam precipitate and then the actual settled layer, and how you analyze them separately. You did some pretty interesting evaluations here.
It is significant. It was the most involved application of electrochemistry that I have done. I've done some other with rainwater analysis that was pretty revealing as well. But this was the first time this level of work had been done with the instrumentation. And if you look at the papers that I wrote on this, once again, it's that thread of history. But what happened was, I do the work, and this was the motive for using electrochemistry. It was to do that the observation transformation of blood was a complete surprise in between. But this was the actual goal. that I started with was determining the chemical constituents the best that I could. What's interesting to me is that if you will take each one of these items that are listed here and you'll look at my paper, you'll see what I'm doing. I'm asking the question, have I ever seen that before? Have I ever seen that before? Have I ever seen that before? And if you go through those papers, you'll see almost, with I think only one exception that I know of actually, but you will find, mild way of saying it would be several. Sometimes it could be up to 24 papers of previous research papers that were identifying these same types of components. I remember some of the earliest work I did in the laboratory notes. I was talking about halogens. This is going back 15, 20 years. I'm saying it looks like there's halogens here. And then I start looking into that chemistry and what does that mean, aromatic substitution, this type of thing. So there is a continuous record of the existence of interest in these constituents going back and embedded in dozens and dozens of papers. And the value of this paper was, it was the first time it was a concentrated effort by let's say a core methodology that said, this is what I'm seeing, this is what I'm seeing, this is what I'm seeing. And it wasn't along my learning curve of trying to ask the questions all the time. Is it such and such? Is it such and such with respect to functional group analysis? So this was a very succinct, rather sort of fundamental identification of constituents that, if we look at it, they actually have a very deeply embedded history and the iron thing is something that has dozens of papers alone written on it, on the iron issue in blood.
Yes. The next paper that you wrote, and by the way, I'm going to link all of your papers and obviously your website so people can study more, because this is work of years and years, meticulous. But this particular paper for me is extremely important, because if we are considering that we might be facing an extinction level event potentially that is facing humanity, because we are being bombarded and assaulted by weaponized geoengineering, by being sprayed with these structures that are basically changing the human body. It's being injected, and it's in the food supply, it's in the water supply. There's now evidence that we find it in all kinds of environmental things like masks, like PCR tests, etc. So my concern is, what can turn this on? And that's one of the questions that David Nixon and Matt Taylor, the engineer, and Shimon Yanovitz, another engineer, we were all asking ourselves. And we definitely found that Wi-Fi makes these structures grow. We're very concerned about light sensitivity, because they appear to be optical communication highways between these microchips that are developing in the vials and these ribbons that we are finding in human blood. And here, what you put together was a potential enlistment of the sources of current or what you would call growth stimulus to this. And you talk about the human body itself. Harald Kautzwala, with whom I'm going to speak here in a couple of weeks, he talks about that biophotonic light of the human body is actually potentially actually making this growth possible as well. But you mentioned magnetic fields, which I'm very concerned about, because a lot of doctors put vaccinated people in MRI scans. I have heard of people who've had very severe side effects, other electromagnetic fields. And you're even talking about environmental things like cyclotronic resonance. Can you just talk a little bit more about some of the larger aspects of how our planet is affected by its electromagnetic field, and how that might be contributing to the growth of these things?
Sure. Thank you very much. This was a paper that I never actually envisioned writing this paper. This paper was not in the plan. as the paper series was unfolding to me, I was going to go from directly what is now 4 to 6, 4 to 6 being the chemical results, and saying, okay, what does that mean? That was the plan that was evolving. What happened was when I reached the end of the chemical, you have two things that happen. So you have a combination of two things. Number one, you have chemical constituent identification, but you also have the mechanism that appears that was never anticipated whatsoever, that causes a complete transformation of your very sample, namely blood, to begin with. So you have this transformation that takes place that was never anticipated. So when I got to the point of wanting to write the last paper and be done, basically, because it was a 9-month project, I stood back and said, well, wait a minute, there's a rather important question here that needs to be addressed. And that is, here you have this case where somebody is showing that you apply some electric current and really wild things happen, dangerous things, and quite potentially lethal things happen to the human body. And so I realized somebody could say, well, that's interesting, but how the hell do I know that's ever going to happen? Just because you recorded it. So that's interesting. But I had the question that had to arise. Well, if it was shown that it could be done, then you must ask the question, are there ways around us already that are in place? Are there existing mechanisms and methods in place in our environment that might produce exactly the same circumstances that I had observed? Now, one of the big problems here was that the current involved here was extraordinarily low. Okay. It's on the order of 1 to 2 milliamps, and that's in a diluted sample. Therefore, in a full sample, we may well be dealing with microamps or microamps. Sure. I'm at the milliamp level. Okay, so this is not very much current. So then you ask the question, do we have environmental forces around us that might produce current at this level? And so I just started listing, and it starts out with a list of 3 or 4, and then the list goes to 6, and then it gets to 12, and eventually it gets to 14. And what I'm realizing is, well, this list can probably be 100, it could be 200. I remember right away after I left it off, I remember thinking, oh my goodness, you got electronic devices all around you all the time going on. And yeah, I started, you know what, I did pick it up in number 13, but the list can go on and on and on, but I had to stop because the list is pretty representative. It's a fair list. It's a fair list of saying, asking the question, are there sources of electrical energy around us that might be able to produce the conditions that were observed? And the answer is yes, in numerous ways. It's not a matter of just it might happen, in numerous ways. And actually, it's expected when you go through this list, you can pick any one of them. And it's more than sufficient to allow for the induction of an effect like I observed. Reading of the human body thing, like athletes, I think the number, like athletes can produce, like it's up to 1,000 or 2,000 watts. When I forget I think the normal range is like 100 watts or whatever, 70. It's still significant, by the way. But an athlete, by the way, it's a curious question about the, let's say, possible increased lethal mortality of athletes. That's an interesting question. But nevertheless, that the body itself, we could have several hours, right, just going through this, but you can pick any single one of these topics. And I would contend that they allow, that's why I wrote them, they allow for the very real prospect of recreating exactly the same type of conditions that were recorded that did transform the blood. That's what this list is about.
And I think it's a very, very important list. And I just wanted to say what you said about the athletes. That's been my concern as well, is the exercise actually that is increasing the electrical conductivity in the body, putting them at higher risk for potentially growing these structures. this was a very interesting thing. Obviously I went through your notebooks and I found all kinds of things, but I just inserted this because you did an analysis that showed that this is mainly a polyacrylic organometallic halogenated aromatic amine. So I'm going to just translate that real quick for people, because polycyclic means this is a carbon-based structure in a hexagonal shape like the benzene rings. And halogenated are these compounds like your bromine, iodine, chlorine, fluoride. And then metallic means all of these metals that we saw, like aluminum, barium, aromatic amines. So these are again type of polymers that are part of synthetic biology. And I pulled this review article just to let people know that polycyclic aromatic amines here are carcinogens. So they're found in things like tobacco smokes, emission from heated cooking oil, but they are mutagenic, carcinogenic, and hematotoxic. And the reason why this is very important is they damage DNA. And I just wanted to show that this can have pretty profound effects on the human body. And I wanted to mention this was Mike Adams' evaluation of the clots, and he found that these are conductive metals as something highly unusual that was chemically reacting with combustible, sparking, popping, fiery reaction. And that are not made of blood. They attract different types of animals more than blood would, and it's a self-assembling carbon-rich structure. So similar to our hydrogel kind of chemical. I always like to give people a little hope, and this is what I found in your in your research, that you looked at different components, which is part of the experiments that in our team, the international research conglomerate, we're also doing. But you found that vitamin C and A, C, and glutathione are actually growth inhibitors for these things. And I do believe that EDTA is definitely a key component, but I think that this is very, very interesting research. I just wanted to mention it. And this is part of the structures that you've also found. But I wanted to spend a little bit of time, Clifford, kind of as we're wrapping up, talking about this particular aspect of it, which is I'm worried about it, and I think that you're worried about it too. Is there a kill switch to this thing? Which means that if someone turns on 5G globally, or if we are exposed to large quantities of radiation, electromagnetic frequency, etc., could this cause a global extinction level event? And I want you to speak to that, because you were one of the first people who really shared my same concern. This is why I'm driven to be out there and talk. I certainly do not care to be known or to be public or anything like that, but I have such an unease about what I know in regards to what is in the vials, what's been found in the blood. Can you say a little bit about this sixth paper that you wrote?
I regard myself as generally of a reserved style, I guess I would say. I'm really not after attention. I'm after very serious things taking place in the world, but I'm not after attention. I actually don't like to make a scene. I like good work to be done, and I like us to be responsible and accountable and do the right thing. That's what I want. But in sort of the true vein of scientific assessment, I guess I would say, when I wrote these papers, not knowing really where was this going to lead to, remember, I never predicted that transformation at all. I was just after trying to find out, why is the blood acting differently? But you have to deal with the reality of what it is that you find and that you see. And we are human beings. You know, this idea of separation of science where you make it, you know, autonomous and machine-like, and the human, you know, if you read the literature of the 1800s of scientists, I mean, it's beautiful. It's absolutely beautiful sometimes what they write, because they're human beings, and it's a part of the experience. So you can do your work to keep things separate, but you're a human being, and a part of the contribution is our humanity. It's not just facts and figures. So when I look at this work, I have to just sit back, and you have to ask, what does this mean? Okay. Go ahead and get involved in the science. That needs to be done. But what does it mean ultimately? And what it meant was what I said in the 6th paper. It's not something, I mean, it was written from a point of human consciousness and human conscience. Okay. And that is, take it for the facts. If you have a situation where the facts portray that they show that the blood of the human being can be transformed in, you know, you could say elementary, but it's not elementary. It's obviously highly complex, but in what can be described in an elementary fashion, namely the induction of electrical energy, electromagnetic energy. If you show that you have a transformation that can take place within human blood by the application of a low-level electric current in a relatively short period of time, to where it brought me to the point of saying, this is not really even recognizable as blood anymore. I can't call this blood. So that means you created something different. So if all of this happens in a relatively short period of time through a relatively elementary method, and again, electromagnetics is complicated, but nevertheless, what does this mean? And what it means, whether I like it or not, what it means is that a mechanism, the means to potentially, I'll go ahead and be diplomatic and say potentially, but you have the means to potentially annihilate the human species.
Yes.
In a relatively brief time. It's conceivable from a physical and conceptual standpoint that it can be done.
Absolutely.
Now,
I agree with you.
and so I had to write it as a fact. As a matter of fact, this was the easiest paper for me to write, because it was so straightforward in terms of what is the impact of all this. I have 50 pages on telling you how such and such was done, but I can tell you in a sentence or two what it potentially might mean. And I did it, and then I tried to be gentler. I mean, some people freaked out. I've had people criticize me for, you know, giving them doom in the world, you know, something that's not easy for them to get out of. That's not the standards of my work. I go where the work needs to go, whatever that is. And I tried to be gentler in the last half of the paper and say, well, you know, it doesn't mean we have to kill everybody, doesn't have to die right away at one time. But guess what? The mechanism for, call it selective, I use the term selective decimation. A method of selective decimation exists. That means it can be applied to what? Any individual, any group, any region. Basically, you can do anything you want or need to do, you being the collective forces, whatever that would be in this case. So you can take this anywhere you want, from the standpoint of, oh, your health might be affected and life will go on, to the fact that, you know, you just might have the sudden death that's never going to be explained. But guess what? It could be explained if you were willing to pursue it. To having a group of people that maybe there's a group of people that are not quite so desirable because they're raising a little bit of heck about something. And, you know, maybe I could apply this method to those people, and you know what? They would never know again really what it is. And so you have a continuum whereby the entire race of humanity, and by the way, it's not just human beings. You'll see my work. I have a paper called The New Biology. That paper is about life on the planet. But the end conclusion was, whether I like it or not, I'm obligated to write what I see as being the consequences of this work. And the consequences are, we have something in place that is in place. This is not something hypothetical that might happen. The system appears to be in place. The evidence says the system is in place. And my question always to the human race is, what would you like to do? What is your level of responsibility as a human being? That's my question. It's always been there.
And thank you so much, Clifford, for all of this. And I agree with your conclusion. This is why I, as a medical doctor, as unpopular as it is, and as much as we're being attacked from other doctors who think that we're seeing cholesterol and salt and, all of this is nothing. And it's like, no, the reason why I continue to be out there, and I'm so grateful for individuals like you, is that this has been weaponized. It is now in all of us. There is no vaxxed or unvaxxed. It's been sprayed on people. It is shedding. It is transmissible. It's on our entire atmosphere. And this can be accessed and be utilized as a weapon via these different systems. And I think that people need to wake up. I am a huge proponent to find therapeutics like EDTA that appears to be working. There needs to be a lot more research, but people need to stand up and demand answers and fight for the survival of the human species. As far as I'm concerned, there's so many critics. I don't have time for these idiots. In closing, in this post-Christmas day, I want to thank you, Clifford, for everything you've done for humanity, for the human species, as a singular, just open-minded researcher. And where now I can go back and others can go back and look at this phenomenal library of work and really find answers to what we're seeing now. You have like 2 decades on us in the fight. I appreciate you. I think you're a hero to humanity. I know that you're a humble man and that was never in your intent, but whatever appreciation and gratitude you've never received, I hope you know that you receive it from me. And thank you so much for speaking to me today. I think that we were able to make some connections for people. And hopefully we'll talk again, because there's a lot to uncover here. And I will also link your website links. Do you have anything else here in closing that you want to mention to us? It's getting dark where you are.
I know I want to apologize for the dark screen, but I don't have a light that's bright enough to bring me into existence, I guess. So I'll do the best I can. But anyway, I want to thank you in return for the same. And I think that I don't believe things happen just totally by accident in a lot of ways. And it's taken us a while to meet, but I'm glad that we have. It's highly significant to me. And it's another stage in the process. And I actually, even though it might seem pretty tough for people, I'm really not a doom and gloom person. And as a matter of fact, that's why I wrote 400 papers. I write out of responsibility, but I think I actually have a core spirit that probably is as, let's say, forward-looking as it can be. And so you don't look back and say, that's the way it is. You recognize and learn about the world around you, and you join in, you dig in. It's like, this is our world. This is not something about, somebody has to do something to me and I'm just the recipient of it. This is our world. Okay. And so it's our chance, it's our choice. The story's not over, and no matter what happens, the story is never actually over. So I guess I also would like to say that even though I don't have a laboratory setting anymore, my life has changed. I'm older now. The work was recorded, and that was that's helpful. It's very necessary and helpful. The laboratory notebooks, as I mentioned to you, they were never intended to be released. They are extremely difficult to read, but I know they're there. And if it really comes to it, I can still read them. At this point, at least, I can still read them. So I do offer myself. I can't replicate what I've done now. I don't have the means or facilities, and my life is different. But we have a record of work, and you and I hopefully have put things in context where we know where we might go to look for things. And then the work, the whole work was done with the point of it being able to be replicated. So everything should be able to be replicated. If not, then that opens up its own questions. But I'm available as long as I'm here to assist in the process of, let's say, interpret. If you can't read my handwriting, let me know. And we go after it, because that's why they're there. It wasn't written to be released, but guess what? It is released. That's another story in itself as to why they were released, but they have been released, and I can still read them. And to me, it's totally logical. I mean, if I had the resources and assets of the CDC, and I could have said this 20 years ago, I know exactly what I want to do. It's all there, okay? And you start by getting core information about what's going on. Get the information, and then use your head. Use your head, and that's the value of the human being, to put it together and figure out where you want to go with this. And believe me, there are plenty of places to go. So let's play catch up if we can. I don't know how much I'll see in my lifetime or not, but let's play catch up, put the piece together. But most importantly, join in, participate, and make your stake, make your claim in this world, because that's why you're here.
I love it. Thank you so much, Clifford.
Thank you, Dr. Anna, and we carry on. And you know what I said, if somebody can't read my writing, I'm there.
Thank you.